Quidel's been a diagnostic company for over 30 years. We were founded in 1979, located in sunny San Diego, California. For the most of the years, we've actually been a lateral flow diagnostic company. Initially with pregnancy assays, and then later infectious diseases with our Strep A tests, and then Influenza A and B. In 2010, we acquired diagnostic hybrids, and in doing so, broadened our platform bases to cellular-based assays, such as DFA, and as well as started our molecular initiatives. And in 2011, we were very happy to have our first molecular assays, flu A and B, and HMPV FDA cleared. So when we started developing our molecular tests, we looked at what was out there and available, both in Europe as well as in the United States, and found that there were some challenges with some of the assays that were out there. And we separated those challenges into workflow challenges and performance challenges. And so we found that many of the kits required you to have frozen reagents, that you needed a storage condition of -70 or -20, which many labs do not have a lot of these facilities available. In addition, you needed ice and cooling blocks during the setup, reagent and control mixing, aliquoting and storing tubes and having to match your batch sizes. Many times there were different cycling conditions for all of the different assays. And so even if you were running a variety of different respiratory assays, because they use different cycling conditions, you couldn't batch them together. Some of the cycling conditions were also greater than 2 hours. Many of these kits also lacked internal controls to control for inhibition throughout the entire process. So what we sought to do was to solve some of these issues when we developed our own molecular PCR kits. So we have refrigerated storage, 2 to 8 degree storage for 24 months from the date of manufacture. We have room temperature setup, so this doesn't require any ice or cooling blocks. You can set it up on your bench top. We have streamlined setup. Everything comes in the kit ready to use. We've got lyophilized master mix, common setup conditions, common workflows, multi-well multiplexing, so that all of our assays that are from the respiratory viruses all run under exactly the same conditions, which means you can choose to run a flu A and B assay alongside an RSV HMPV assay. In addition, we've sought to have short assay times, so about 70 minutes for the respiratory virus assays, and about 60 minutes or under for some of our C. diff and other assays. We use common pipetting steps and common setups so that once you're trained on one assay, it's a similar procedure for all of them. We also use a common internal control for both our RNA and DNA-based assays. And then we have broad equipment validation. So we have clinical data from 2 real-time PCR platforms, soon to be 3 with the QuantStudio, and we validated our assays in a wide variety of other PCR platforms. In addition, we get 510(k) clearance on our platform, and all of our manufacturing is done under GMP and ISO 1345 conditions. So the real-time PCR assay kit configuration, it's a 96 reaction kit with lyophilized master mix. The master mix is provided in 12 vials with 8 reactions apiece. There's also 1 vial of rehydration solution, 1 vial of internal control, and this is all shipped on blue ice. Again, 2 to 8 degree storage and 24 months shelf life. So very long shelf life, which is great for a respiratory season. In case you don't use your entire kit 1 year, it's actually good to use the next. We also have external controls, and these are 2 to 8 degree storage as well. They are stabilized liquid format and single well positive control. So you don't need to have a flu A control and a flu B control. And today I'm introducing the QuantStudio DX and as well as our real-time PCR kits. And the QuantStudio is actually here in Madrid, downstairs at the Life Technologies booth, so if anyone wants to check it out in person. And why do I care about influenza? So the first assay I'm going to talk about today is the Quidel molecular real-time flu A and B assay. But first, why do I even want to test for influenza? So as you all know, influenza is an RNA virus that causes illness characterized by fever, chills, headache, sore throat, coughing, runny nose. There are 3 subtypes of influenza, influenza A, B, and C. Influenza A and B are the primary human varieties of influenza that cause disease. Influenza can also be confused with a wide variety of other infections, such as the common cold, rhinovirus, adenovirus, para, as well as other bacterial infections, such as strep A, strep pneumonia, and chlamydia pneumonia. Globally, influenza infects about 3 to 5 million people every year, and also kills between 250,000 to 500,000 in non-epidemic years. it's very important to test for influenza for a variety of reasons. First, if influenza is diagnosed within the first 48 hours, there are treatments available. There are a couple of drugs, such as Tamiflu and Relenza, that are effective against both flu A and B strains. And also, because influenza is an aggressive illness, and like I mentioned, kills between 250 to 500,000 people, it's necessary to identify very quickly whether someone has flu or a simple cold. So the Quidel molecular flu A and B assay is a real-time PCR kit for the qualitative detection and identification of both influenza A and influenza B, from nasal swabs, nasopharyngeal swabs, aspirates, and washes. The product is right now cleared, FDA cleared, on the Life Technologies 7500 Fast DX system, as well as the Cepheid Smart Cycler 2, in conjunction with the bioMérieux EasyMag extraction system. We've also validated the performance on the entire suite of Life Technologies 7500, as well as the Roche Light Cycler 480, and the assay is currently being validated on the QuantStudio DX platform. So the workflow for the Quidel molecular respiratory assays it's the same regardless of the assay being used. The kit supplies you with the process control, the master mix and rehydration solution. The end user supplies themselves with the ABI qPCR plate, the specimen, and the EasyMag sample strips. The middle line shows you the extraction procedure, 5 simple steps. We take 20 microliters of the process control and add that to each sample well. You then take 180 microliters of your specimen, add that as well. You initiate the onboard lysis procedure on the EasyMag. You add 100 microliters of the silica beads, and then initiate binding and purification. At this point, you are left with 50 microliters of extracted specimen. And this extracted specimen can be used for both influenza A and B, as well as RSV, HMPV, and some of the other respiratory assays that we're developing. So for the amplification and detection procedure, it's also 5 simple steps. You rehydrate the master mix, so you add 135 microliters per tube of master mix to be rehydrated. You pipette it up and down a couple of times, and that's it. You then aliquot 15 microliters of that master mix for every specimen you are going to test, and then 5 microliters of your extracted sample. You then put the plate into the 7500 Fast DX system, or the QuantStudio system, initiate thermal cycling, and an hour and a half later, you get your results. So we have some performance data. Initially, this assay was FDA cleared, like I said, on the 7500 and the Cepheid Smart Cycler. This slide details the clinical trial data from our 2010 respiratory clinical trial that we performed. In comparison with a competitive PCR kit, so commercially available FDA cleared real-time PCR kit, we found that we were able to pick up 8 additional flu A specimens, and these flu A specimens were positive for flu A by sequence analysis. And interestingly, 26 out of the 28 flu B specimens were also positive by sequencing. So we identified 5% more flu A positives and 21% more influenza B positives than a comparator kit. In addition, the comparator kit had about 2-fold more inhibition in specimens, so yielding invalid results, than our kit. We had less than 1% inhibition rate, and that's with over 700 specimens evaluated with this trial. We've now undertaken some preliminary clinical specimen testing, comparing the performance on the ABI 7500 Fast DX to the QuantStudio. We'll be initiating a clinical trial with the QuantStudio this fall as respiratory season starts. But the initial studies have shown that there's 100% comparison, both sensitivity and specificity, with the initial 166 specimens that we evaluated on both platforms. And so we feel very confident that the QuantStudio is going to have equivalent, excellent performance as the 7500 does. We also did a series of analytical studies. We evaluated 38 different influenza A strains. That includes the pandemic California strain, a couple of the other pandemic strains, seasonal H1 strains, seasonal H3 strains. In addition to all of the strains that are highlighted in red are strains that are derived from animals. So we have the H5N1 avian influenza strain, all the way from H2 down to H16. every single different subtype of influenza that we've evaluated has been correctly identified by this assay. In addition, we looked at a variety, 13 different influenza B subtypes, and found that we were able to detect all of those as well. So we feel that we're very confident in the analytical reactivity for this assay. We've also done some cross-reactivity testing. We looked at a panel of different viruses, and we also looked at some bacteria that were found, that could be found in a specimen, and found that we had no cross-reactivity with anything that we looked at. So anything from RSV, HMPV, adenovirus, EBV, BKV, HIV, there was no cross-reactivity with our primers and probes. In addition, we also looked at interfering substances, so anything that could be found in a specimen, including blood, mucus, some nasal sprays, throat lozenges, and importantly, some of the drugs that are commonly used to treat influenza also had no interference at 3x or 10x, the limited detection of the assay. And finally, we did a performance study looking at multiple different media types. So what we found here is in blue is flu A, red is flu B. We looked at different transport media from UTM and saline to the different M's, so M4 and M4RT, M5 and M6. And we found that there was very little difference in performance whatsoever in terms of measuring the average CT for any of these different transport media types, so that we have broad capabilities of accepting and testing any specimen regardless of the media transport type. So now switching gears a little bit, we're talking about the new RSV HMPV assay. The intended use for this kit is for the qualitative detection and identification of RSV and HMPV viral RNA. We have done clinical specimen validation on the 7500 FastDX and Smart Cycler 2 and extracted samples on the EasyMag. Now this assay, we just finished our initial clinical trial and have submitted to the FDA. The specimen types are nasal swabs and nasopharyngeal swabs, aspirates, and wash specimens. So all of the respiratory assays will be tested on the same set of specimen types. And we're also in the process of validating this assay on the QuantStudio DX. And the clinical trials for both the HMPV RSV assay as well as the flu A and B assay will be running simultaneously. And so what's important to note is that, as I mentioned the workflow before, the extracted samples can be used with both the flu A and B assay as well as the RSV HMPV assay. They use the same thermal cycling conditions, and so they can be run on the same plates, side by side. If labs choose to run a flu A and B, they can then run a RSV HMPV right next to you. This is a summary of the clinical trial data for RSV HMPV. In comparison to the Pro HMPV kit from Prodessa, GenPro, we found that there was excellent positive and negative agreement. We have about 97% positive agreement with the Prodessa kit and 99% negative agreement. All of the originally discordant specimens, the 3 additional HMPV positives that we picked up, were bidirectionally sequenced and found to be 2 positives. And with RSV, we compared this to DSFA and cell culture with DFA, found that we picked up an additional 21 positives that DFA missed, and all of those positive specimens were true positives by bidirectional sequencing. And the negatives were actually false negatives by sequencing. And so we found that our sensitivity and specificity were about 98% for this assay. We did a variety of different analytical tests, so analytical specificity, inclusivity. We evaluated the 2 different subtypes of RSV in a variety of different strains. In addition, we tested a variety of different clinical isolates. We found excellent performance on all subtypes tested. We did this for HMPV as well. There are 2 major subtypes of HMPV. There's A1 and there's the A and B, and A1 and B1, as well as A2 and B2. We sourced a variety of different strains from different locations and evaluated them all, determined the LODs, and compared on both platforms and found excellent results for both of those. In addition, on the right-hand side, we did the same cross-reactivity panel and found that there was no cross-reactivity for 12, let's see, there's like 15 different strains of viruses. We also tested a variety of different bacterial. So those were the viral assays that we are currently evaluating on the QuantStudio. I'm going to switch gears a little bit and talk about our first assay that we'll be launching next month, which is the toxigenic C. difficile assay. And C. difficile, while I realize this is a viral symposium, is a bacteria, but it's been in the news lately and of great interest. So C. diff is a diarrheal disease. It can cause life-threatening inflammation of the colon, and actually can kill quite a few people every year. In the US, they just published some statistics. There are about 30,000 deaths due to C. difficile in the past year. This illness occurs most frequently in older adults who are hospitalized and have been on antibiotics. So after antibiotic treatment, this wipes out your intestinal gut flora. You can then have a C. difficile infection. One of the issues with this is a hospital-acquired infection. It's very challenging to get rid of C. diff in a hospital. Patients who have C. diff excrete between 1 times 10 to the 4th and 1 times 10 to the 7th spores per gram of feces. And these spores are particularly virulent in that they live on surfaces and can only be cleaned with bleach. In the US, this is estimated to generate over $1 billion in excess healthcare costs annually. It also increases the patient length of stay by up to 7 days and increased cost of inpatient care for more than $7,000 per episode of C. diff. We have developed a molecular assay that is direct, so it does not require automated extraction. It's a sample to answer in less than 60 minutes. The kit provides you with a sample prep kit that uses a neonatal sampling swab, process buffer 1 and process buffer 2. And then the PCR kit provides the lyophilized master mix and the rehydration solution. The process buffer 2 actually contains the internal control. The direct stool sample preparation is as follows. You collect the sample using a supplied swab, dipping it into solid or unformed stool. You then transfer this into the first process buffer. You twirl the swab by rolling the handle between your index finger and thumb. You then take 30 microliters of the initial PB1 into the process buffer 2. You mix by pipetting up and down, and that's it. No time steps, no vortexing. That's the lysis process. You then rehydrate the master mix, adding 135 microliters of the rehydration solution to the lyophilized mix, 15 microliters into each well, just like the respiratory assays, and then 5 microliters of processed specimen into each sample. You then insert the plate into the 7500 or the QuantStudio and initiate thermal cycling. The thermal cycling conditions are less than 60 minutes. And so in 1 hour approximately, you get your results. We've done some initial studies now comparing to both cytotoxic cell culture or another CE-marked IVD molecular device, and found that with cytotoxic cell culture, 7 out of the 8 additional specimens that we picked up were positive. And we missed 1, that was invalid in the other device. So we had an initial positive percent agreement or sensitivity of 94% and an initial specificity of 93%. When compared to another FDA-cleared and CE-marked molecular device, we had excellent agreement, 96% positive agreement and 99% negative agreement. We also evaluated the assay against an enzyme immunoassay. This study was done by one of our collaborators, Dr. Kent Mitchell, down at CPL in Austin, Texas, and found that all 10 of the initially negative specimens by EIA that were picked up by our assay were positive in other, either cell culture or another FDA-cleared device. And the 2 specimens that were positive by EIA and negative by our molecular assay were also negative by both cell culture cytotoxin as well as the other FDA-cleared device. So we had excellent positive percent agreement and negative percent agreement with this test. We've also done some very initial clinical specimen testing on the QuantStudio DX platform and found that our initial sensitivity and specificity were in the 90s in comparison to cytotoxic cell culture. And 9 out of the 10 that we picked up in addition, in comparison to cytotoxic cell culture, were also positive by BD genome assay. And then both of these negative specimens on the Quidel molecular C. diff were negative by both the BD genome as well as the Cepheid gene expert assay. So there's about 91% agreement between the cytotoxic cell culture and the C. diff assay. And the corrected sensitivity and specificity when all of the discordants were resolved were 100%. So this assay is excellent performance on the QuantStudio. We're going to be initiating our clinical trials starting next week on the QuantStudio platform, so we're actually pretty excited about that. In addition, we've done a variety of different testing on different C. diff ribotypes and toxinotypes, and looked at both the 3B as well as toxinotype 0 in terms of LOD. And our assay LOD is less than 1 CFU per assay. And I believe. And finally, coming soon, we're actually in the process of developing a variety of other assays for both the QuantStudio and the 7500. We've got the HSV 1 and 2 VZV direct assay, and this will be another extractionless-based assay. We have an adenovirus and para-influenza 1, 2, 3, and 4 that will use the same respiratory workflow, so these assays can be run in conjunction with either flu A and B, as well as RSV HMPV. And we're also developing a Bordetella pertussis parapertussis assay. And finally, I'd like to thank everyone at Life Technologies, including John, Travis, and Kim, and then my large Quidel Corporation team for actually performing all of this work.